Hello everyone, so we're going to tackle bioinformatics report on the protein ubiquitin. Our group composes of Yui Chua, Vivian Xiao, and Bea Sablan. So staying virtually unchanged throughout the course of evolution, ubiquitin is a small 76 amino acid regulatory protein that is present in all eukaryotic cells, especially in all the cellular tissues of humans. Through directing movement of significant proteins in the cell and participating in both synthesis of new proteins and destruction of defective proteins, ubiquitin plays a major role in numerous cellular processes in relation to the regulation of proteins. So, ubiquitination. So, this is a process when a ubiquitin molecule binds to a substrate protein and will undergo a type post-translational modification. So technically, it attaches two proteins and tags them for disposal. The tag proteins are then taken to proteasomes to be destroyed. This is a three-step enzymatic process. First is by the activation with ubiquitin activating enzymes, followed by conjugation with ubiquitin conjugating enzymes, and lastly is by ligation with ubiquitin ligases. The ubiquitin forms an isopeptide bond with a residue of lysine on the protein substrate respectively. So it may also occur by way of a peptide bond at the end terminus. The substrates may either be modified by addition of a single ubiquitin molecule, or also called as monoubiquitination, or by the conjugation of ubiquitin to preceding ubiquitin moieties, or also called as polyubiquitination. So there are various results of ubiquitination, and as stated earlier, the most common process is by degradation of protein by proteasome. However, it may also be affected by the activity, location, or interaction with other proteins. So it can affect cell division and multiplication, the generation of neurons and muscular cells, DNA transcription and repair, immune and inflammatory response, receptor modulation, stress response pathway, and viral infection. So now we move on to the cofactors. So the AAA, ATPase, CDC48, P97, and VCP is deemed to participate in the post-ubiquitination step by facilitating the processing of substrates. While this enzyme can bind ubiquitin, it necessitates various cofactors such as UFDI NPL4 heterodimer to effectively process protein substrates and act as ubiquitin receptors in ubiquitin fusion degradation pathways or UFD pathways. So, importance of ubiquitin function in drug discovery. So yes, this gene is present in all eukaryotic organisms, especially in all the cellular tissues of humans. So based on its function, studies regarding ubiquitin have been conducted as it may have a role in potential targeted therapy to treat cancer. So the main idea is that ubiquitin will be used to manipulate protein in cancer cells to cause the cancer cell to die. So studies regarding ubiquitin, approved by the Food and Drug Administration or the FDA, led to the development of three proteasome inhibitors that treat people with multiple myeloma, a form of blood cancer. So these are bortezomib, carfilzomib.
Next is the BLAST-N of ubiquitin. Shown here are the results of the BLAST-N interface. Particularly, we will be dealing with the UBB or ubiquitin B of humans, which is indicated by the red box. So the group has chosen the first results of BLAST-N in humans since it exhibits the highest score and an expected value of 0. This means that this nucleotide sequence shows the most significant alignment match. So this shows the GenBank data of the blast and output, and the GenBank accession number is NM018955. From this, we could see that the UBB gene has 17 chromosomes and it starts at base 1 and ends at base 933. The translated sequence below will be used for the FASTA sequence of BLAST-P. So here are the 933 nucleotides of UBB. Next is the BLAST-P of ubiquitin. Similar to the previous output, the results with the most significant alignment is the polyubiquitin B precursor for Homo sapiens. And after clicking the alignments tab, it shows that there are 229 amino acids on the protein polyubiquitin B precursor. So the GenBank data has an accession number of NP00128645. Its summary indicates that the protein ubiquitin is synthesized as a precursor protein consisting of either polyubiquitin chains or single ubiquitin moiety fused to an unrelated protein. This gene consists of three direct repeats of the ubiquitin coding sequence with no spacer sequence. Consequently, the protein is expressed as a polyubiquitin precursor with the final amino acid after the last repeat. With its 229 total amino acids, the three different sites of the protein that exhibits the function on interacting with activating enzymes will be found in the position 54 and 72 of the protein sequence, and its essential function is found on the position 68. So shown is the protein sequence with its 229 amino acids. And then the MAT peptide indicates the first repeat of the ubiquitin coding sequence consisting of a peptide of 76 amino acids. At the same time, the position 65 indicates the site for phosphorylation in the polyubiquitin protein. And then the other two repeats of the peptide of 76 amino acids is shown here. After the last repeat, which is around 200, which is 153 to 228, the protein is expressed as a polyubiquitin chain with its final amino acid at position 229. Next is the protein structure. And from here, we could see the general properties of ubiquitin with its molecular weight, 8,579 grams per mole, with its 76 amino acids, and its isoelectric point Pi is at the value 6.79. So the protein ubiquitin is one of the most conserved proteins known, and from this we could see the homologous genes of UBB in different species.
We will now examine the 3D structure of a particular protein. Among the four results from the NCBI BLAST protein of the amino acid sequence of ubiquitin B, only the protein TNIP2 UB complex in C2 form exhibit a three-dimensional structure with a PDB ID number of 5H07. However, the gene name of this molecule is UBC. There are some features that must be taken into account. This 3D structure consists of helices and also beta sheets. The helices are formed by amino acid residues that are highlighted in pink. Then, for the beta sheets, it is also formed by the corresponding amino acid residues. Then, the accessible protein residues are primarily found on surfaces of this protein, helix or beta strands. Then, for the aromatic rings or side chains that are bulky, it is predominantly found in the core of the protein or in tight turns of the protein. These are the amino acid residues that are buried within the protein core. Lastly, there are some disulfide bridges located between cysteine residues, as you can see here. After inspecting the structure of 5H07 in PDB, it is discovered that this protein is classified as signaling protein in Homo sapiens with E. coli as its expression system. It is also evident that there is no mutation present in the protein. In order to visualize this structure, X-ray diffraction is employed with a resolution of 2.59 Armstrongs. Using the structure classes of proteins database classification, the protein contains a mixture of alpha helices and beta sheets, which is indicated by class. Its fold is considered as beta grasp, which is predominantly found in ubiquitin. As such, its superfamily is attributed to ubiquitin-like as it is closely related to ubiquitin. Scientists have also uncovered that this protein does not bind with ligands. First relevant macromolecule similar to the protein 5H07 is polyubiquitin C, particularly chain A with 228 amino acid residues in Homo sapiens as well. This molecule is also recognized by its ability to tag proteins through covalent interactions, particularly via isopeptide bonds among lysine residues, which eventually forms polyubiquitin chains. Aside from polyubiquitin C, 5H07 contains two peptide chains of TNF-AIP3 interacting protein 2 with the gene symbol of TNIP2 with a length of 91. This second macromolecule also engages in protein binding, ion binding, enzyme binding, and kinase binding. As such, it regulates the stability and structural integrity of the protein and contributes to various biological regulation processes. The total molecular weight of 5H07 is 47.13 kilodaltons. Thank you very much for listening.